Let me introduce our next speaker. We've got Chunyang Zhang from Indiana University. She's going to come and talk to us about mitochondrial and nuclear incompatibility during oogenesis results in embryonic lethality. All right, just give us one moment. All right.
Okay. Good afternoon, everyone. First of all, I'd like to thank the organizer for giving me this opportunity to present our work related to human mitochondrial disease. Okay. So we know that mitochondrial dysfunctions are highly relevant to a series of human disease. A lot of this disease, such as aging, obesity, diabetes, and cancer, has become major health concerns in modern society. And noticeably, current studies suggest that there's a strong correlation between the quality and the quantity of mitochondria with female fertility. So as the power plant inside cells, mitochondria has its own genome. However, this small genome only encodes 37 gene products. In great contrast, mitochondria actually harbors more than 1,500 proteins. This means that the majority of the proteins has to come from nuclear gene products. And at the same time, also, this put a great emphasis on the compatibility between these two genomes. Whether Mito Nuclear incompatibility can lead to any human disease is still not clear in human. Today, I'm going to introduce you a Drosophila Mito Nuclear incompatible model. These flies were initially created by our collaborator, Dr. Christine Montus, and her colleagues when she was working in David Rand's lab. So we know that Drosophila melanogaster and simulans are two different Drosophila species. And within melanogaster, Austria and Oregon are two naturally existing populations. By outcrossing among these flies, they were able to generate mitonucleic hybrid flies. Essentially, we could have mitochondrial genome in a different nuclear background. For example, Sim Austria means that it has a simulans mitochondria in the melanogaster Austrian nuclear background. So when they examined the phenotype of these flies, they found that 3 of them were pretty normal, except one, the same organ flies. These flies shows incompatibility between these two genomes, indicated by the developmental delay, reduced muscular functions, and the temperature-dependent infertility. And this very last phenotype became very interesting to us, and it will be the main focus of today's talk. So molecularly, our collaborators also traced back to the two genomes. What they found was the incompatibility actually lies between two genes, one encoded by the simulans mitochondria and the other one encoded by the organ nuclear genome. So in simulans, there is a polymorphism in the mitochondrial tyrosine tRNA, and at the same time, in the organ nuclear background, there is a mutation of the mitochondrial tRNA synthetase. To remind you, what the tRNA synthetase does is to charge the tRNA with the corresponding amino acid. So if you take a close look at these two genomes, there is a polymorphism in the mitochondrial tyrosine tRNA, changing a GC to GU pair. At the same time, in the organ nuclear background, there's a mutation, changing a very conserved alanine residue into a valine. To remind you, this alanine is not only conserved in Drosophila, but it's also conserved all the way through human being. It only got changed to a V residue in the organ nuclear background. So coming back to the infertility phenotype, essentially, we were not able to get any viable offsprings from the stock that was raised at a higher temperature, which is the non-permissive temperature at 28 degrees. Then in order to eliminate the male effect, we crossed these females with wild-type yellow-white males. Still, we were not able to get any viable offsprings. So the question became, how does Mito Nuclear incompatibility cause female infertility? So to answer that, firstly, we look at oogenesis process. Here, we collected G0 embryos, either keep them at 25 degrees or shift them up to non-permissive temperature for further development. Then we collected newly eclosed G1 adult females, cross them to yellow-white males. Here, we're analyzing the egg production rate of these different females. So on the left, we have females raised at permissive temperature, and on the right, non-permissive. And these 4 different colors shows you the 4 strains we're analyzing here. To remind you, both of the organ strains has a V allele in the tyrosine tRNA synthetase. You can tell that the black strain, which is our incompatible strain, has a lower egg production rate under both conditions. However, this is not quite distinguishable between the incompatible strain with our compatible organar strain. We have also looked at oogenesis process. To remind you, Drosophila egg chambers are generated from germarium, where the germline stem cells are residing. And at the anterior tip of the germarium, we have 2 germline stem cells. And with mitotic division, germline stem cells will generate germline stem cell daughters. And here, I'm using an antibody called HATS1B1 to label the germline stem cells. In a normal-looking germarium, you are expecting to see 2 spherical structures at the anterior tip of the germarium, telling you where the germline stem cells are. However, in the incompatible strain, we're getting a very high frequency of losing this structure, suggesting defective germline stem cell maintenance. And here's the quantification. The gray bar shows you the germline stem cell maintenance is normal, and the white and black bar shows you defective germline stem cell maintenance, indicated by loss of germline stem cells. And you can tell that under non-permissive temperature, we have a very high frequency of loss of germline stem cells. However, this is also true in our compatible organar strain, although this phenotype was enhanced in the incompatible strain. And using cell death markers, I have also found that there's a very high frequency of germline stem cell deaths and stem cell daughter deaths. So previously, it's been shown that if you challenge the germline stem cells with irradiation stress, germline stem cells are actually resistant to apoptosis. My results suggest that germline stem cells are actually competent for apoptosis if you challenge them with metabolic stress. To summarize this part, what I just told you was both strains with an organ nuclear background has loss of germline stem cell and stem cell daughters. And this phenotype was enhanced by the Mito Nuclear incompatibility. However, our incompatible strain was still able to generate normal-looking egg chambers, although we were not able to get any viable offspring. Then our next question became, what is the real basis for this incompatible strain-specific infertility? So next, we decided to look at what happened to the next generation, what happens to those embryos that were being generated. So similarly, here we collected newly eclosed G0 adult females and crossed them to yellow-white males. Here we're looking at the hatch rate of G1 embryos. From this figure, you can tell that in the incompatible strain, the embryos from these females has a much lower hatch rate compared to other compatible strains. And to remind you, the black strain is our incompatible strain. And I have also done rescue experiment. My rescue experiment results suggest that one copy of the compatible allele of the Adam gene or tyrosine tRNA synthetase was sufficient to suppress the embryonic lethality phenotype, suggesting that phenotype is indeed caused by the incompatibility between these 2 genes. Next, we wonder whether the temperature-sensitive period is in the mother or in the embryos. To answer that, we allow these G1 embryos to develop either at permissive or non-permissive temperature. From this figure, you can tell that it is actually the developmental temperature of the mother that determines the hatch rate of the embryos. So based on this, we conclude that there is a temperature-sensitive maternal-effect embryonic lethality raised from this Mito Nuclear incompatibility. So our next question became whether this maternal effect is reversible if we switch the adult females back to permissive temperature. So again, we collected newly eclosed G0 adult females, cross them to yellow-white males, and shift them back to permissive temperature to analyze the hatch rate of next generation. And here's the result. And black line is our switchback experiment. You can tell that it actually indeed shows a reversal effect. However, it takes a very long time. It only shows this reversal effect after 8 days of temperature shift. This makes us wonder why it takes such a long time to reverse the effect. So if you take a look at the timeline of oogenesis, it actually takes about 8 to 10 days to generate a mature egg all the way from germline stem cells in the germarium. So this leads us to wonder whether the temperature-sensitive period in the mother actually lies in the germarium. To test this idea, we decided to go back to the germarium to see what happens there. So here we're using an ATP5A to label the mitochondrial morphology in the germline cells located in the germarium. And using super-resolution microscopy, what I found was in the compatible strain, mitochondria shows this smaller or more individualized structure in the germline cells. In contrast, in the incompatible strain in the germarium, mitochondria shows this tubular or even aggregate structure. At this point, we still don't understand how does the mitochondrial morphology in germarium dictate the hatch rate of the embryos. So to summarize my talk today with a model, in the compatible strain where you have an A allele of the tyrosine tRNA synthetase, we get normal germline stem cell maintenance, and the germline stem cell daughters can survive. And at the same time, we have a smaller mitochondrial structure in the germline cells in the germarium. As a result, we can produce more embryos, and these embryos can survive. In contrast, in the incompatible strain, where we have a V allele of tyrosine tRNA synthetase with the incompatible tyrosine tRNA, more often we get germline stem cell loss and loss of stem cell daughters. At the same time, mitochondria shows tubular or aggregate structure in the germarium, and put this together, we get fewer embryos being produced, and these embryos will eventually die due to embryonic lethality. And as I said, at this point, we still don't quite understand how does this influence the survival of the next generation. But we have two non-mutually exclusive models. We think it's either a temperature-sensitive period or a temperature-sensitive process in the germarium, or there's a certain kind of metabolic memory or even epigenetic memory that's being set up in the germarium that dictates the hatch rate of the G1 embryos. So we're currently in collaboration with Dr. Jason Tennyson to test this idea. Now coming back to the disease model, there are a lot of mitochondrial tRNA synthetase mutation has been correlated to human disease. More specifically, YARS2, mutation in YARS2, which is a human ortholog of our tyrosine tRNA synthetase in fly, has been linked to myopathy and some other disease. However, our results suggest that certain allele of this YARS2 mutation may also cause female infertility. And more broadly, our result also should suggest that Mito Nuclear incompatibility can also lead to female infertility. With that, I would like to thank my graduate advisor, Dr. Brian Kelvey, and all other Kelvey Lab members. And we thank our collaborators for the very helpful discussions. And we thank all the IU facility and reagents and funding resource. With that, I'm happy to take any further questions.
I was wondering if you know that the mutated tRNA synthetase actually goes to the mitochondria, whether it's imported and localizes to the mitochondria properly.
So we haven't looked at the tyrosine tRNA synthetase location yet, but based on the prediction, it does have a mitochondrial localization signal.
So you don't think it's because the tRNA synthetase doesn't go to the mitochondria?
It's a good question. We don't know yet. We haven't looked at mutant yet.
Great, Doc. I wonder how general this is. Have you thought about that? Is this also the potential reason for interspecies reproductive barrier?
I think this could be one of the major reasons. But I think this is also very rare because the other hybrid flies, the same ostrich, does not have the same problem. So I think in order to create, there is a very low frequency to create this molecular incompatibility.
Well, great. Thank you so much.